Hello viewers, today I will discuss the semi-conservative DNA replication. Okay? The complementary relationship between adenine and thymine and cytosine and guanine residues ensures that the bases are aligned in the right manner and the sequence of bases in the daughter DNA molecules is exactly the same as in the parent molecule. With the result, the two double helices produced are identical to the original one so that the information content remains unchanged. And this type of replication is known as semi-conservative replication. Okay? So, in this kind of replication, the DNA polymerases are required. Okay? The basic enzyme is DNA polymerase. The E. coli cell contains 3 different types of polymerases, that is 1, 2 and 3. The DNA polymerase 1 discovered by Arthur Kornberg in E. coli is a monomer which catalyzes the addition of the D NTPs to the 3' end of the double-stranded DNA. Its single polypeptide chain has about 1000 amino acid residues. All 4 deoxyribonucleoside phosphates are required for its activity. Catalytic reactions required Mg2+ or Mn2+ and a primer which is known as RNA primer. Okay? And the primer have free 3' OH end. Hydrolysis of the PPI drives the 5' to 3' synthesis. About 20 phosphodiester bonds are made in one stretch, after which the enzyme is knocked off the DNA template. Apart from polymerizing reaction in 5' to 3' direction, the DNA polymerase 1 has a proofreading activity. It has exonuclease activity in the 3' to 5' and 5' to 3' directions. But 3' to 5' exonuclease activity reads the result of a polymerization cycle. Next one is DNA polymerase 2. DNA polymerase 2 was discovered after 15 years of DNA polymerase 1 discovery. It has a molecular weight of 120 kD and possesses 3' to 5' exonuclease activity. The enzyme is unable to replicate long single strands with a short complementary primer and possibly carries synthesis in 5' to 3' direction. Biological functions of DNA polymerase 2 are not yet fully known, but it has this kind of activities. Moving to the DNA polymerase 3. It is by far the most active of the 3 polymerases. It is 15 times more active than DNA polymerase 1 and 300 times more active than DNA polymerase 2. It is multi-subunit enzyme and carries all the activities present in the DNA polymerase 1. It has a molecular weight of about 140 kD and has about the same template primer dependence as DNA polymerase 2. DNA polymerase 3 normally occurs in a more complex but biologically active form which is known as DNA polymerase 3 and is believed to be a dimer of DNA polymerase 3. DNA polymerase 3 requires an auxiliary protein. Evidence has been provided by Arthur Kornberg and his colleagues that DNA polymerase 3 combines with DNA polymerase copolymerase 3